Hi everyone, welcome to Pathology. So today we are moving on with an interesting topic. Before that, do you know that your own body cells will eat themselves to survive? Have you heard of it? It's scary, right? So it's nothing but the term autophagy. So today we are going to learn about autophagy for both undergraduate levels and also in the postgraduate level. So I'm here to explain about the key concepts for the autophagy, what are the types of autophagy and how they affect in a normal development and also in a disease. Okay, so we'll break down the term autophagy. So auto in the Greek refers to a self and phagy means eating. So it's basically self-eating. So the cell is saying I'm fine, I'll eat myself for the survival. That's all. Okay, so the mechanism by which the cell consumes its own component. So the cell will break down itself and consume its own component for the survival of the same. Okay, so here they are providing an example of aging and exercise. Ever heard that a fasting or exercise will slow down your aging? Yes, right. But do you know the mechanism what's behind it? That's a thing called as autophagy. Okay, so here the cell starts to clean up your body like a refreshing button. When you do exercise or starvation, it will eat itself and try to, it's like a biological refreshing button. That's all. Okay, so in 2016, yes, this self-eating tiny mechanism had won a Nobel Prize by Yoshimori. So the scientists have explained about the mechanism of autophagy and got the Nobel Prize. Alright. So what do you do when your room is messy? Definitely we'll try to clean it, right? We'll remove all the debris and then we'll try to do the same thing. The same thing happens in your body cells. Okay, so that mechanism is known as autophagy. So here coming to the functions of autophagy. So there are three main functions. So first one is to degrade and recycle your intracellular content. So I told you an example, right? So whenever your room is very dirt, you will try to clean it. The same thing is happening within your body also. So whenever there is a starvation or something happening in your body, some stress is happening, the cell will try to eat itself for the survival. So it will degrade and it will recycle the intracellular content. So that is one of the most important mechanism of autophagy. So second one is to remove the damaged proteins and organelles. So here what it is true. So to keep the things fresh and also the functional, it maintains the normal homeostasis. So to maintain the normal homeostasis, the cell is turning over the autophagy button. So next coming to the other functions, like apart from cleaning and whatever the function, it also has some other function where it will clear the misfolded and damaged protein, and it also has a host defense mechanism, and it protects from the tumor process. Also it protects from neurodegenerative diseases. So these are the main functions of autophagy. So you should remember what is autophagy and what are the functions of autophagy. So next, moving on to the categories. So there are three main types. One is macroautophagy, the next one is microautophagy, and the third one is chaperone mediated autophagy. So you should remember all these three types. It's a very important. So by the term only you can understand what is the concept behind it. Macro means it's a little size bigger, right? Micro means it's smaller, and chaperone means it's a kind of something which is helping in autophagy. That's all. So we'll see about each. Okay, so the first one is macroautophagy. So there are major steps in it. So macroautophagy means first what will happen, there will be initiation. So once the cell understood there is some trouble happening outside, okay, like for example as you told, exercise, starvation and aging, any damage, okay, it's like the start button. So that is your initiation step. So then the cell starts its nucleation and elongation with the isolation of membrane. So that is nothing called as a phagophore. So phagophore is nothing but it's like a small bubble like element. Okay, membrane will be formed outside the cell. From the cytoplasm, it will form like a projection. It's called as a phagophore. Okay. So next, the third step is autophagosome. So autophagosome is nothing but, so now the bag is sealed with the autophagosome. Okay, so you think that a double membrane vesicle will form like a bulge. It's carrying all the trash. So remember the initiation is like whenever the trash is in your room or something you are going to put in a washing machine, like a start button is your initiation. So next it's preparing or it's collecting all the trash, that is your phagophore. Okay, so the next one is it's sealing all the trash, that is your vesicle. Okay, and the fourth step is it fuses with the lysosome. Okay, once it fuses with the lysosome, it forms a component called as autophagolysosome. Okay, this autophagolysosome is a key element where it starts the process. Okay, so here now the real digestion starts here. So the lysosomal enzymes within the cell will try to digest it. Okay, and then it will break down all the cell. The cell has a cytoplasm and a nucleus, right? So this autophagolysosome will activate and it breaks down all the cellular components. So within the nucleus you have mitochondria and everything is there, right? So all these things will be break down. Then it will leave a residual body. What is residual body? Once you clean the whole area, it will be either totally left or there will be some small elements, right? So those are called as a residual body. So once the residual body has a two fate, either it should be completely removed or it should be left over. So the leftover part is called as a wear and tear pigments. So that is your lipofuscin. So these are the characteristic of aging. Okay, this is the whole mechanism of macroautophagy. So to summarize, there will be initiation and phagophore, then vesicle formation, autophagolysosome will happen, then the lysosome will try to break down all the cellular components, and finally it will leave the residual body and it will go for either completely removal or wear and tear pigments called as lipofuscin, and this is the characteristic of aging. So this is the whole mechanism of macroautophagy. Okay. So next one is you are moving on to molecular basis of macroautophagy. So if you see in this diagram, you can see there is an initiation complex, then comes your nucleation, and the nucleation autophagolysosome is formed, and the next one will be your lysosomal digestion, and finally it leaves a residual body, and that will be engulfed either completely removal or the wear and tear pigments will be formed. Okay, so next moving on to the molecular basis. So here if you take the molecular basis, okay, so meet the commanders. These are the commanders for cleaning process. So that is your ATGs, autophagic genes. So that can be like 5, 7, 12, 16, etc. There are a lot of numbers which are responsible like a checkpoints for the mechanism of autophagy, which will lead to initiate the process. So once these are the components which is like a gate reservoir, they are required for formation of autophagosomes. Once this autophagosome is formed, the ATGs are responsible for your initiation button. So once the initiation is done, then it will lead to nucleation, and then nucleation after that it will go for elongation, and later it will activate your lipid phosphatidylethanolaminine, LC3 will be activated, and finally there will be autophagolysosomes and there will be degradation. So this is the molecular basis which is responsible for the gatekeeper is your autophagolysosome genes. Okay, that is your ATGs. That is the main thing which is responsible for initiation of the process, and finally it will lead to degradation. So the next one is microautophagy. So you think of lysosome as a little monster inside the cell. So we saw macroautophagy where there will be initiation, nucleation and autophagolysosome component, and finally the lysosome will digest the cell, right? So next moving on to microautophagy. So this is something happening within the cell. So that's why it's called as microautophagy. So here everything happened within the cell. You think that there is a little monster inside the cell. Okay, it's sitting inside and waiting for some of the cytoplasmic elements. Okay, snacks kind of thing. So instead of waiting and for the delivery of the trash, it's directly acting for the target and then it's killing the concept. Okay, so that is known as a microautophagy. So this happens like through the invagination, like a pocket. Okay, the lysosome will fold inwards the cell within the cytoplasm, and it's like justly it's like, you know, the man munching dots like a game. You saw the Pac-Man game, so it's something like that. Okay, Pac-Man game. So where the cell eats its own content, like some of the nearby thing. So it's the same kind of thing which is happening in the microautophagy. So here the cytosolic targets are directly ingested by the invagination of the lysosomal membrane. So your cell has the cell membrane, right? So this cell membrane will invaginate along with the lysosome and forms like a pocket. So they are transferred interior into the cell degradation. So once there is a stress or something, all the damages elements will come inside and fall inside that pocket which is made by the cell. So then it starts, lysosome is already present in the membrane, so it doesn't need any initiation, nucleation or something like that, nothing is needed. So it will start the turnover of the membrane and it will start for cellular degeneration. So this mechanism is known as microautophagy. So how will you differentiate macroautophagy and microautophagy? Macroautophagy, the cell will build like a bag, like autophagolysosome vesicle is formed and then it will start the digestion. Okay, so it actually happens like a stepwise process. So if you come for microautophagy, here the lysosome eats directly the target. Okay, so no bag, no waiting, just the cell, whatever the damages comes, and the lysosome will form a pocket and it will be engulfed by the thing and the digestion happens. So that's the difference between macroautophagy and microautophagy. So next, coming to what is the action of microautophagy. So the main action is they directly engulf the cytosol by lysosome. Okay, so the lysosome is the one which will form like a pocket, so that will be engulfed by that. What is the mechanism behind it? The lysosomal membrane invagination. And how they use us for recycling and energy saving during the starvation. What is the key difference? There is no autophagolysosome formation here. There is only invagination, direct action. Okay. What is the clinical relevance? Aging, cancer and neurodegeneration, everything will happen. So you can refer this diagram for all those microautophagy and macroautophagy difference. So next comes your chaperone mediated autophagy. So here you can see the diagram over here. So there is a cell cytoplasm with LAMP2A, LMP2A, and there is a chaperone which you can mention as C, and there is particular protein outside the cytoplasm, right? So we'll explain this diagram a little later. So come to chaperone mediated autophagy. Here there will be a direct recognition by the heat shock protein, whatever it is, there will be a direct recognition. There will be a specific target or a protein which is responsible. So the chaperone and the protein will bind. Now this chaperone protein will walks into the cell, that is like a lysosome. So the lysosome LAMP2A is a security door, which is very key game for this chaperone. So here the chaperone acts like a passcode. So once the chaperone acts like a passcode and your chaperone along with the protein walks into the lysosome, so the passcode is delivered, then it is verified by your LAMP2A. That will helps the target protein to slide within the lysosome. So once this action is done, there is no vesicle, no autophagolysosomes, nothing is needed here. This is like a direct engulfment. Okay, that is a key concept of chaperone mediated autophagy. So the protein, specific protein is there, specific chaperone is there. So they both interact, and along with the LAMP2A is a gate reservoir where it acts like a key and it will try to react with the lysosome. So this protein and chaperone is forming a complex, and this complex will be coming into the cell. Okay, so here your LAMP2A is a gate reservoir. So that is a passcode. So once the passcode is checked out, your chaperone and protein complex will react directly with the lysosome and it will go for degeneration. This is the simple process happening in the chaperone mediated autophagy. Okay. So next, moving on to the autophagy and disease. How they affect and forms the disease. Okay, so it has a very significant implication, as you see, in from the development stage of the baby to the death, everything it has a significance. So it's a main role in the aging process and also the cancer development and neurodegenerative diseases like Alzheimer's and Huntington disease. So furthermore, the autophagy is implicated also in the infectious conditions like HIV, Shigella, Mycobacteria and inflammatory bowel disease. So we'll see about it everything later. So it's actually speaking like a multi-phased role. Okay, it has a multi-phased role both in health and the disease, right? So it has an important of understanding autophagy for potential therapeutic interventions. What they are trying to say, it's very important what kind of autophagy and what level of gene is affected in particular disease, so that you can give a targeted therapy. So that's what they're trying to say here. So diseases linked with the defective autophagy. So if you take Parkinson, it's basically the impact mitophagy. What is mitophagy? It's something the autophagy happening in mitochondrial level is called as mitophagy. So if there is a defect in mitochondrial dysfunction, that will ultimately impair your mitophagy. Once the mitophagy is defect, then all the unwanted proteins will be deposited in your brain. It will affect your brain. Obviously, it's known as a Parkinson's disease. It's a neurodegenerative disease where the unfolded proteins will be presented. Okay. So next, coming to the cancer. So you all know during starvation, the autophagy starts, which will be further prevent the steps of cancer. So if there is a autophagy defect, it will ultimately guides towards your tumor progression. And the next one is infection. So here, all the many infections like HIV and all of those things, they block xenography. Once the autophagy is blocked, then the pathogen will lead to survival. Okay, so that's how it has a mechanism behind that. So Crohn's disease, you can name ATG16L1 mutation. So this is the autophagy responsible for inflammatory bowel disease. So if there is a failure in this particular gene, it will lead to Crohn's disease. That's what they are trying to. It's a new recent advances where they have detected this type of autophagy gene is main cause for Crohn's disease. So next, coming to the therapeutic implication of autophagy modulation. There are three: inducer, inhibitor and natural enhancer. So examples are rapamycin, chloroquine and fasting and chloride. Effect are mainly they promote the autophagy or block the autophagy or enhance the autophagy. So what are the therapeutic application for aging, cancer research, and also sensitize the tumors on chemotherapy, longevity studies. So next, coming to the comparison, summarizing here. So what is the difference between macroautophagy, microautophagy and chaperone mediated autophagy? So microautophagy, there will be no autophagal lysosomes. So autophagal lysosome will be present only in macroautophagy, where it will be not present in both, that is micro or chaperone mediated. There is no autophagal lysosome. Target specificity is moderate in microautophagy because the specific protein should come. And if you take macroautophagy, it's moderate to selective. The particular target should come, then autophagal lysosome should activate, then the lysosome process will be happening. And the third one is chaperone mediated. So this is highly specific. The particular chaperone should go and bind to particular protein, then it activates your LAMP2A as a passcode, then finally it goes for direct lysosomal destruction. So that is your highly sensitive one, is your chaperone mediated autophagy. And the energy and machinery needed, the minimal energy is needed for microautophagy and chaperone mediated. Whereas the complex machinery involvement will be involved in your macroautophagy. It usually occurs at, the micro will occur at lysosomal surface. I told you microautophagy is nothing but the lysosome, the cytoplasm will have invagination like a pocket coated with lysosome. So it's like a lysosome and the surface will be present in microautophagy. When you come to macroautophagy, the cytoplasm and also the lysosome will react in the macroautophagy level. And in chaperone mediated, same, lysosomal and your cytosomal thing will be acting. But here the most concept is your receptor. LAMP2A receptor is a passcode for this. So I hope so you will be able to understand the concept now. Difference between macro, micro and chaperone. So macro is like a big process where you have nucleation, elongation, and you have autophagal lysosome complex, and finally there will be a degradation. Once the degradation occurs, it will leave your residual body. The residual body will be either completely resolved or it will go for wear and tear pigments called as lipofuscin. So this is all about macroautophagy. Microautophagy is nothing but, I told you, there will be a cytoplasmic invagination coated with lysosome. So directly all the stress and cytoplasmic organelle will come inside the pocket and go for destruction. That is your microautophagy. So the third one is chaperone mediated autophagy. So chaperone mediated autophagy is nothing but there will be a formation of the chaperone and the protein complex. So once this complex comes in, along with the passcode of LAMP2A, it will be activating the lysosome and your protein chaperone complex will be reacting and it starts the degeneration. So this is the basic mechanism between all these three. To summarize, you have to remember all these key points. In your exam, you can draw along with the diagrams and everything. So I hope so you people understood a little concept regarding autophagy. So we'll see in the next class with some other new concept. Until then, bye bye.